Greetings to all. I am Dr. Ali C. Anthony, Assistant Professor, Department of Biosciences, MES College Marampally, Kerala, India. Respected dignitaries and delegates, first of all, let me thank all the organizers of the Global Conference on Polymers, Plastics and Composites, PPC 2021, for giving me an opportunity to speak in front of this wonderful audience on my topic, fecal contamination and prevalence of pathogenic E. coli in shellfish growing areas along southwest coast of India. As we all know, food security is one of the most important issues that world faces today. Fisheries and aquaculture are important sources of food, income and livelihood for millions of people around the world. Among seafood, the category bivalves are important source of proteins, vitamins, minerals, essential fatty acids, and they contain less saturated fatty acids and also provide protection from coronary heart diseases. But unfortunately, the disappointing fact is that there are a number of reports worldwide regarding outbreak of several diseases associated with shellfish consumption, and hence it is often considered as a potential health hazard to the consumers. These are a few reports on the outbreak of diseases associated with shellfish consumption, and the main reason is the contamination of the water bodies in which these bivalves survive. Most of the shellfish growing areas are situated in shallow near shore waters which often receives sewage from all sources, and they are also rich in nutrients. The sanitary quality of shellfish growing areas are often found not to be satisfactory. And these bivalves, they are in fact sedentary organisms, and hence they are highly prone for contamination, and they have a peculiar non-selective filter feeding nature also, as a result of which they tend to accumulate almost all the contaminants, chemicals as well as pathogens present in the surrounding water. As a result of which it is found that the increased levels of fecal contamination and pathogens are often found in these bivalves, and this poses potential health hazard to the consumers, and often many disease outbreaks are associated with shellfish consumption. Another important reason that aggravates this problem is the traditional way of consuming this shellfish. In many parts of the world, it is often consumed raw or partially cooked or undercooked. And that is the reason why Center for Disease Control and Prevention (CDC) has classified this group of seafood, that is bivalves, under foods that can cause food poisoning. They have also issued a number of directives in this regard to control the outbreak of or contracting shellfish consumption associated diseases. The main source of data are CDC, WHO and FAO reports, and according to these reports, seafood is listed high on in the list of foods that are capable of transmitting food associated diseases. And it is estimated that as per FAO reports 2014, 10% of all the foodborne disease outbreaks are somehow related to seafood. But the fact is that only less than 1% of the true incidence is actually reported, that is under-reporting exists. And most of the available reports are from US and Europe. From Asia and Africa, very less reporting. Japan has got good surveillance and reporting. In developing countries, there is lack of surveillance and no much data is available. As per CDC reports in 2013, 818 foodborne disease outbreaks were reported, out of which 11% was related to mollusks. And in these outbreaks, about 34% was due to Salmonella pathogen and 7% was caused by Shiga toxin producing E. coli. Among the diarrheal diseases that are found associated with shellfish consumption, the main causative was enteropathogenic E. coli and followed by enterotoxigenic E. coli. In this slide, you can see some of the symptoms of diarrheal diseases associated with E. coli: diarrhea, abdominal cramps, bloody urine, then loss of appetite, etc. Then because of this concern, there are a number of shellfish sanitation programs by international agencies in order to regulate or ensure good sanitary quality of the shellfish growing areas. Two important such agencies are National Shellfish Sanitation Program NSSP of US FDA and European Council EU regulations. So, these international agencies insist certification of the growing areas based on fecal coliforms level or E. coli, and they classify the areas into approved, restricted, then prohibited, etc. If the sanitary quality is found to be unsatisfactory, such harvesting areas are closed for harvesting. But such regulations exist only in such European or US. In most developing countries, there is no such regulations or survey by such agencies. We have conducted a study in this regard on fecal contamination and prevalence of pathogenic E. coli in shellfish growing areas along southwest coast of India. The study area that was selected was Cochin Estuary, the biggest estuary located towards the southwest coast of India. And this estuary was selected because it has very good impact on the local population. Thousands of local men depend on this estuary for their livelihood, and the seafood exported from this estuary is exported worldwide. Then another important reason is this estuary, it has undergone increasing levels of fecal contamination as well as other contamination recently, and the reason is there are a number of satellite townships present bound on the banks of this estuary. There are also a number of seafood factories, hospitals, domestic dwellings, etc. on the banks of this estuary, and these are some of the photographs of the sampling sites. The samples were collected early in the morning. The samples selected included sediments, harvesting waters, and live Indian black clams. The scientific name is Vilorita cyprinoides variety cochinensis. It is endemic to the study area, and season-wise sampling was conducted for a period of 1 year from January 2013 to December 2013. Pre-monsoon, monsoon as well as post-monsoon sampling was done. Identification and isolation of fecal coliforms as well as E. coli was done as per standard microbiological methods prescribed by APHA, 3-tube MPN method, Invictus, molecular identification of E. coli by PCR based detection of species specific UIDA gene. This was followed by serotyping of E. coli, which was done at National Salmonella and Escherichia Center, Central Research Institute, Kasauli, Himachal Pradesh, India. We also investigated the presence of toxigenic genes in E. coli by multiplex PCR described by Payton et al. 1998. The toxigenic genes investigated included Shiga toxin genes XTX1, STX2, intimin gene EAE, enterohemolysin gene HLYA. So, the presence of these toxigenic genes will reveal or will give us a better picture about the actual toxigenic or pathogenic potential of the isolated strains. That was the intention of investigating the toxigenic genes. We found that seasonal variation existed in the prevalence of fecal coliforms in shellfish, sediment and water. It was found that high level of fecal contamination was found during monsoon and post-monsoon seasons when compared with pre-monsoon season. It was also found that shellfish and sediment samples exhibited higher levels of fecal coliforms and pathogenic E. coli compared to water samples. We could identify 17 diverse serogroups of E. coli among the E. coli strains isolated from all the samples isolated from Cochin Estuary, which included both pathogenic as well as non-pathogenic strains. It was found that about 89% of the total E. coli isolated from all the samples together were pathogenic, and we could identify 5 diverse pathogenic serogroups of E. coli, and among them enterotoxigenic E. coli prevailed, showed the maximum prevalence, followed by enterohemorrhagic E. coli and uropathogenic E. coli. Coming to sample wise distribution of pathogenic E. coli groups, it was observed that in shellfish Vilorita cyprinoides, 84% of the strains that were isolated from shellfish was pathogenic, whereas from sediments 78% were pathogenic, and in case of harvesting water 69% of the strains isolated were pathogenic. This slide shows the presence of toxigenic genes in E. coli. Out of the 4 toxigenic genes investigated, all the 4 toxigenic genes were present in the samples isolated, and this slide shows the gel images of the products of the toxigenic genes investigated. So, these toxigenic genes, these are actually responsible for the diarrheal disease that is caused in human beings. The intimin gene, it helps to attach the pathogenic cells to the intestinal mucosa. Then the Shiga toxin genes STX1 and STX2, it will help to inhibit protein synthesis in the host cells as well as kill the cells, lysis of the cells, and the androhemolysin gene, it will lead to hemolysis of the RBC. So, bloody urine is sometimes found in severe cases. This slide shows the relative prevalence of toxigenic genes in various samples as well as coexistence of various toxigenic genes in various samples. So, we can see that the maximum prevalence was shown by the toxigenic gene STX2. This was followed by toxigenic gene EAE, and lesser prevalence was shown by STX1 and least prevalence was shown by HLY-A. From this we can see that the prevalence of high prevalence, about 78.1% showed the prevalence of toxigenic genes, and the strains isolated exhibit high virulence potential. Conclusions: high levels of fecal coliforms were detected in monsoon and post-monsoon season from the study area. Similar results are reported from various parts of the world, and this is a matter of concern because high levels of pathogenic E. coli in sediments may act as repositories for future contamination of the overlying water when some hydrological events such as upwelling happens, because these sediments, they offer a very favorable environment for the survival of microorganisms, because it is a nutrient rich environment, then the temperature is very favorable. So, they tend to exist or survive for a prolonged period, and when the upwelling events or when some disturbances happens, it may contaminate the overlaying water. So, again recontamination of the aquaculture products may happen. Then shellfish and sediment demonstrated higher fecal coliform levels compared to harvesting waters. Then majority of the E. coli serogroups were pathogenic. They also exhibited higher levels of presence of toxigenic genes. That means the virulence potential is also high. Then this is the first report on the isolation of E. coli 0157 from shellfish harvesting areas along Indian coast. So, present study reveals poor sanitary quality of the harvesting area as well as potential health hazard posed to the consumers. Then way forward, these are some of the suggestions from our study to mitigate this issue. So, we suggest regulatory intervention should be implemented to improve the sanitary quality of the harvesting areas, and regular monitoring should be done for the presence of pathogens such as E. coli 0157H7 or some other emerging pathogens or salmonella etc. should be implemented on a regular basis. Then the shellfish harvested should be subjected to some purification processes such as depuration or subjected to thorough cooking. So, in this slide you can see the image of a depuration system that is developed in our lab based on a recirculation water system with activated charcoal filter. This was found to be effective in purifying clams isolated from the estuary. So, with this let me conclude. Thank you all for your patient hearing. Thank you.